Many thanks to the organisers for the opportunity to come to a real live meeting with real live people. I'm going to try and introduce UK Health Security Agency to you. I'm a scientific leader for healthcare biotechnology based at Port and Down, and I'm also a professor of antimicrobial therapy based at King's College London. So, as you might imagine, for an organisation in the public health space that came into existence during a global pandemic, I think these mission statements and aims are rather fluid. So this was a document that was published back in March. UKHSA came into existence as an executive agency of the Department of Health in April, and we've been fully operational since October. I'm happy to provide the slides if anybody wants to read this, but this is kind of the high-level mission statement of what we're trying to do. A lot of it's driven by data around genomics, and I think with a very heavy emphasis at the moment, as you might imagine, around COVID. So I'm not going to labour this too much because I think this will change over the next 6 months into something that reflects a maybe sort of broader public health landscape. So this organisation has been in existence slightly longer. So this is UKHSA Port and Down. We're all now part of the Research and Evaluation Division of UKHSA. And we have a long tradition in translational biology, supporting translation of therapeutics, drugs, diagnostics, really around high containment microbiology and drug-resistant pathogens. That's the real focus of the centre. So all of these things so everything we do has an applied purpose, but that doesn't mean to say that we don't do important underpinning research to understand infection processes, pathogenesis, immune responses, etc. We do a lot of work on discovery and evaluation and efficacy testing for therapeutics and vaccines, and microbiology transmission studies, environmental microbiology studies, looking at decontamination and environmental detection, and as you might imagine, quite a lot of work on diagnostics. Thank you. Okay, if anybody can't hear me at the back, please wave. And over the last year, the group's contributed to something like 70 publications, predominantly around COVID. So that gives you a kind of broad landscape of what we do. Porton has a long history of applied infection models. So you can see through the middle here some of the areas that we study, and I've kind of highlighted the ones that are perhaps most relevant to this meeting in terms of immunology, bacteriology, AMR, biofilms, microbiome. But we have a range of non-animal, including insect models, cell culture models for infection, through small animals, NHPs, and a lot of work supporting human trials. And that's been a particular feature of work over the last 2 years. So if I just pick the immunology, I'm going to do a bit of signposting for other people who are here. So I'm not going to labour this too much. Andy Gorringe, Steve Taylor and Steve Thomas are in the audience. I think they're doing a 2-minute pitch in a moment. But I think it's fair to say they're interested in all things to do with immune responses, particularly innate immune responses and development of functional assays. And they already work with a number of partners in the room. I think we're also interested in looking at the interface of the immunology with antimicrobials. Because if you're looking at a new drug for a new antibiotic, it never works in isolation. And I think we don't understand very much about that interface, and I think that's an area we'd be keen to explore. And that kind of leads on to what my group do. So we developed a slightly different outlook a few years ago, because we recognised that PHE at the time, UKHSA now, had a lot of strains and models which were not necessarily freely available in the academic community, and in many cases were hard to access even for SMEs. So we came up with this open innovation concept. So this is where we use those strains and models to support others in the academic community, in the research community. And we work with academics, SMEs. We host a lot of visiting workers. Many of those are not microbiologists. And we provide them with a training in how to evaluate antimicrobials and drugs. Where we sort of add value, I think, is because we have a very strong genomics emphasis and a very strong phenotypic emphasis around those strain panels. It means that the data that people will generate has more meaning in terms of mapping out future development options. So in many cases, which we can show you examples of, there's clear evidence that data has supported future grants and applications in the future. Don't worry about the poster, it's over here if you want to see it in real life. Charlie Hines leads this work, and Charlie can talk you through this. And the kind of little thumbnail sketches at the bottom, you'll see in a moment. So there's a brochure if anybody's interested in seeing this. So this is just showing you a little bit of the brochure, so don't worry too much about the detail. We can give you a copy if you're interested. So what we managed to acquire a few years ago was a large infrastructure grant from the Department of Health and Social Care that built on this original open innovation idea, which is the antimicrobial screening work that happens in my group, but started to look at other ways where we can again leverage skills and expertise within UKHSA to support research in this space. And I'll show you a slide on this in a moment. But these are just some of the kind of case studies that are in the brochure to sort of show you the type of work we do and the type of emphasis we put on terms of these relationships. So there's two messages here, I guess. One around interdisciplinary training. So I say we work with a lot of people who are not traditional microbiologists. And the second one is about driving innovation. And I'm just going to show you one of these case studies in a very little bit of detail, just to give you a flavour of that. So this is technology that was invented by Professor Hal Morgan, Dan Spencer at the University of Southampton. This is the subject of an Invention for Innovation grant by NIHR, which also brings in Professor Reed at Southampton. And what we're looking at doing is developing an evidence-based system for assessing antibiotic susceptibility. So we use this acronym IFAST, impedance-based fast antibiotic susceptibility testing. And what Hal and Dan did is they invented this system where you've got a microfluidic chip. This is a bacteria, a population of bacteria that flow through a series of electrodes, and you put a current across these electrodes and measure the impedance. So essentially, it's how good a resistor is the bacteria in the population as it passes through this system. So the raw data is these kind of frequency diagrams, but what it translates into is this. So these look a little bit like flow cytometry traces, that they're not flow cytometry, they're impedance cytometry traces. And the x-axis is measuring what we call the electrical radius of the cell, and the y-axis is measuring what we call membrane permittivity. So to my simple non-engineering mind, this is how good a resistor the cell is in terms of this measurement. So you can take a population of bacterial cells. This happens to be Klebsiella pneumoniae, but it works for pretty much all the bacteria we've looked at. You can contour that in terms of 80%, 90% of the population. And if you treat that cell population with carbapenem, in this case, and if this is a susceptible population, even within 30 minutes, the entire population moves out of the contour. So this is the basis of this rapid AST test that we're trying to develop. Happy to talk more about this if it's useful, but I think it's a great example of both regional collaboration, but also interdisciplinary collaboration, because this is something that we would not be able to do on our own. Open innovation, there is some money available. That's attracted people in previous talks. So we have seed project funding for studies, particularly looking at non-traditional therapeutics. This is the DH term. To treating multi-drug resistant pathogens. If you're interested, catch either Charlie or myself later, and we're happy to explain more. This is the next bit of signposting. So Ginny Moore is one of my colleagues who leads a section of work on AMR and the environment. Hopefully opening very soon is this facility at Porton. This is a multi-bed hospital ward where you can do a variety of studies looking at colonisation. So in this sense, the sort of things down here, how colonisation changes with use, how you can intervene with that, whether we can see microbiome population movement, horizontal gene transfer, but then on into transmission, infection control, and sort of development of methods and techniques. So please catch Ginny if you can. This is a really fantastic facility, and we're looking to build capability around this in the future. So this open innovation, we were a bit worried, I guess. This is a slightly different way for a research institute to approach things. We wondered how well this would go down. The support from NIHR, DHSC was very useful, but we've got collaborations all around the world in this space at the moment. There's about 60 publications over the last 5 years, and a lot of it's driven by PhD students. So we have 35 PhD students who either completed or are currently in the process of completing their PhDs. and funding from some of the people over on the right-hand side. So this is just very quickly to acknowledge the people in my group who did a lot of the work, but probably more so to highlight this very crude colour coding here. The green ones are chemists, the blue ones are engineers, electrical engineers, physicists. So it's a very multidisciplinary, collaborative environment that we work in, and we're very keen to expand from that because we think it's very important in this space. This is just to acknowledge the funding, particularly from NIHR, which out both for the Open Innovation and for the Invention for Innovation work. And the last slide I'm going to leave you with, the last two slides, is a bit more signposting. So we don't work in isolation of Port, and there are very close links between the different groups. So we're really pleased to be able to work with Joanna Bacon and her TB drug discovery group. Jo has got a core expertise around these kind of chemostat models. So the ability to put a bacteria, this in case TB, into an environment that replicates what's happening in the host. So they can be used to look at drug evaluation, they can be looked at host pathogen interactions, looking at resistance for emergence and also tolerance, not just sort of traditional resistance. Heterogeneous populations, not all bacterial populations are exactly the same, so some really good flow cytometry work at high containment. And then sort of growing areas around rapid high throughput screening of compounds, using these kind of systems as the entry to those kind of models. I'm not going to labour this one because you'll see it again in about half an hour. This is Charlie Hind. So she leads the Open Innovation Programme. Please have a chat to Charlie, have a look at the poster, and if anybody's interested in a brochure, please let us know. And that's me done.